within my group. So actually, in this topic, I'm going to talk about how we can use pHLIP-coated liposomes to deliver most of the hydrophobic drugs and other hydrophobic entities selectively to tumor sites or the cancer cells. So that is the object. And at the same time, I want to mention that this is the fundamental work, and we are doing further studies to translate it into the further beyond this. So before I go into the experimental work, I'd like to mention a couple words about cancer. I think it's advantageous to continue this discussion. So, so people have already identified several hallmarks for cancer. So cancer is, we all know, it's different from the normal cell because of several reasons. One of the significant hallmark is it has reprogrammed energy metabolism or the altered energy metabolism. As we all know, our normal cell, it uses mitochondria to produce most of its energy, we call oxidative phosphorylation. But in cancer, it's a little bit different. So it uses glycolytic pathway mainly to produce its energy. So it's, apparently, it's like energy inefficient, but somehow it uses this pathway. So this is first observed actually 90 years ago, around 90 years ago from today, by Dr. Otto Warburg. So therefore, we call this effect Warburg effect. So what happens is, like in the cancer cells, when they use this glycolytic pathway, it produces byproducts. So lactate is a byproduct. So this can acidify the cell cytoplasm. That's a problem. So even cancer cells or the normal cells, they don't like to disrupt their pH or other ion concentration inside. They want to regulate always the intracellular and extracellular ion balance. So what cancer cell does is, it has adapted to always pump out these extra lactate and the protons. So therefore, surrounding this tumor microenvironment, or fundamentally the cancer cells, we can see like slightly higher acidicity than the normal cells. Because we know extracellular acidicity is pH 7.4, but near the vicinity of the cancer cells, the surface is below that. So especially in microenvironment of tumors, we can see this microacidicity. So this is important for cancer, actually, in many aspects. But for us, when we think about traditional chemotherapy, it's not a good point. Because most of the drugs, traditional drugs, they can't go into the cells and do their job because of this acidicity. And also, it can also help to create drug resistance. So our goal is, can we use this extracellular acidicity, which is a negative thing, as a hallmark to target the cancer cells? And how can we do that? In this discussion, the peptide pHLIP comes to the stage, and it can play this role. So pHLIP we call pH low insertion peptide. Actually, it's a 36 amino acid linear peptide. And as my colleague explained a couple minutes ago, so it has 3 states. So it's mainly a random coil in normal pH, and it's negatively charged. But at the vicinity of the membrane, it can associate with the membrane, but not necessarily it doesn't insert into the membrane. But when you drop the pH, so it can make an alpha helix. So always this formation is unidirectional. C-terminus goes inside. So if you see the especially CD data relevant to these 3 stages, you can see at low pH, so it gives the classic alpha helix formation spectrum, so which is evidence for its alpha helix formation. At the same time, when it forms this alpha helix, it releases energy around 7 kilocalories per mole, which can be used to deliver cell-impermeable cargo into the cell. And this C-terminus can be modified or conjugated with like some cargo, and it can be delivered into the cell. At the same time, the N-terminus can be modified with a flag or a biomarker. So this proves that pHLIP can target acidicity. So next, I'm going to explain about our goal. So what we did in our experiments. So our goal was actually to make holes in cancer cells. So that was the primary idea using pHLIP liposomes. Because if we can make nano holes in the cell surface and other intracellular organelle membranes, so we can actually disrupt the ion balance, and also that could lead cell to go to apoptosis path. So I created, my colleague in my team, we made like pHLIP liposomes with hydrophobic drug, which is actually an antibiotic, gramicidine. So it's a very traditional channel former, and people have studied extensively about this in 1970s, and people know lots of things about this. And we coated this, our liposomes with pHLIP lipids, and as I mentioned, when it comes to the acidic tissues, not only cancer cells, any acidic environment, it can sense this acidicity, and also it can deprotonate aspen glue residues, and it can insert in the cell membrane and fuse with it, or it can take endocytotic pathway and go through fusion inside. So whatever the way, so it can deliver these pores, nanopores, into the cell membranes. So then it can destroy this ion regulation and can create cell death. So let me explain about gramicidine. Actually, gramicidine is one of the first antibiotic available for us commercially. And it's discovered like around like 1939. And it was derived from Bacillus brevis, the bacterium. And it has actually a mixture. That means depending on this Y, like we call it A, B, or C. But in particular, this project, I use gramicidine A to make channels. But the other negative part is, even though this is a very powerful antibiotic, we can't use it internally because it destroys our red blood cells in much faster rate than it kills bacteria. Therefore, this gramicidine, we use it as a topical lotion mainly. So this is actually how gramicidine makes these channels in the cell membrane or the liposome membrane. So usually, when it introduced to lipid membrane, it makes beta helix. So it spans roughly a half of the length of our membrane. And when these beta helices come together, it makes the dimer and it makes the iron 4. So usually this diameter is 45 angstrom, 4 to 5 angstrom, which is a selective channel for all the monovalent cations, including protons. And it usually transfers around 10 million cations per second. So these are facts we know. So next thing was, since we know this fact, we made our liposomes, and we want to see that, okay, people have studied about this. Actually, do they form these channels? So we conducted our biophysical studies for gramicidine-containing liposomes, and we could produce the typical beta helix formation CD graph and also tryptophan fluorescence, and which shows that this forms beta helix. And then what we did was we want to show that, okay, before we move forward, do they actually transfer these protons? What we did was we made like 2 groups of liposomes. One liposome group was with gramicidine, and other liposome group was without gramicidine. And both liposomes were coated, and inside and outside were coated with FITC lipids, which is a pH-sensitive lipid. Then we ran our kinetic studies. So what we did was we dropped pH from 8 to 3, and as you see in this blue line, so even within the dead time of this mixing, like the ones with gammacidine channel, you will see a sudden drop of FITC fluorescence. But without gammacidine, so you will see it takes longer time. So this proves that, yes, it transfers protons through this channel. And then we did our stability studies. And these, according to the stability studies over 1 month in our buffer media, and at 4 degrees Celsius, we could find like it has around 100 nanometer diameter, hydrodynamic diameter, peak diameter. And also it shows with pHLIP, it shows minus 30 millivolts of zeta potential. So that says that the shelf life is quite long, so that we can store them carefully. And then we want to go to our cytotoxicity assays and to see whether this pHLIP-coated gammacidine liposome make any toxicity. So I actually used 3 cell lines. And then what we did was, we treated cells, these cancer cells, at 2 different pHs, because we want to see like whether they do toxicity independent with the pH. Then there is no point. That means it even targets the normal cells. So as you see in these cytotoxicity assays, you will see in gray bars or columns, at pH 7.4, they don't show any cytotoxicity effect with pHLIP. So that means that pHLIP-coated liposome does not affect the normal pH cells. But the ones we treated with at pH 6.2, which is acidic environment, and we see like concentration-dependent toxicity. So then we want to know at what point these cells start their cell death. So we performed ECIS assay, which is a capacitance measurement of cells in microplates. And we found that after 1 day from the treatment, it start dying. So we didn't stop from that point, and we want to see actually how the intracellular pH changes with these gammacidine ion channels. So what we did was, we used a pH-sensitive dye called SNAF. And first, we calibrated for different pHs the cells and made calibrated curves. And then we treated our cells at normal and low pH with gammacidine and pHLIP. And as you see in the second day, these red dots, you will see like the second day of the treatment, the low pH cells, their intracellular pH is around 6.5, while the normal pH cells treated with pHLIP-coated gammacidine, its intracellular pH is staying 7.2, which is normal. So this shows that it changes intracellular pH. And then we haven't stopped from that point. We want to see what happens to other cations, monovalent cations, especially sodium and potassium. So we all know that outside the cell, like the sodium concentration is roughly 150 millimolar. And inside, it's around 5 millimolar. For potassium, it is reverse. So this balance is very important. So as you see here, like you see in this, when we use this corona green indicator for sodium channels, sodium indicator, if there is more accumulation of sodium, so it can give green fluorescence. If not, it's like it doesn't give any fluorescence. You see that in control cells, like even after day 1 and day 3 from the treatment at normal and low pH, you don't see any, or you can see a little green fluorescence. But this group, the panel D, you will see that the treated ones at low pH, so they show much higher green fluorescence. That shows us, that is evidence that so it already disrupts the sodium ion concentration inside cells. So then we went to our third study, that is we want to see okay, if there's a cell death, does this lead to apoptosis? So that's what we did in our next level. So we went to mitochondria depolarization assay, because if the mitochondria is depolarized, that means it is in the path of apoptosis. So we use JC9 dye, and this JC9 dye is an indicator of apoptosis. And if the cells are in good condition, are healthy, so they give more orange color fluorescence. But if they are dying and going through the apoptosis path, then they give more green color. So if you see here, the first, these panels, control panels, you see more orange signal. But with this D group, and which is low pH treated with pHLIP grammacidine liposomes, and day 1, day 2, and day 4, we see this green signal increases while orange signal decreases. So this is a clear indication that mitochondria are all already depolarized and it is going through apoptosis. So this is the preliminary work we did with these entities. And at the end, as a summary, so we can say that acidic tumor, microenvironment of tumors are acidic, and it can be used as a universal targeting method for most of the cancerous tumors. And also, we can use pHLIP technology as a targeting method for these acidic tissues. And in this particular work, actually, pHLIP-coated grammacidine can deliver these channels into our cancer cells, and also it can lead cells to apoptosis. And not only this is like a fundamental study and proving this, that pHLIP can do things. So we can use this technology to deliver lots of other hydrophobic agents like paclitaxel, and also other peptides and proteins and other hydrophobic agents to disease sites. So at the end, I want to thank my team, especially my supervisors, and also I want to thank NIH for giving us grants to continue this project. Thank you.